Genes and the human condition, from behavior to biotechnology. Definition: a gene is a region of DNA that encodes a functional RNA or protein product, and is the molecular unit of heredity.
So, what is genetic engineering? Genetic engineering is when the genetic code of an organism is altered to produce a desired trait. This is done by taking a gene from one and inserting it into the DNA of another to get the wanted end result. To get a better sense of how this is done, let's first understand how DNA works. In any living thing, the DNA acts as the instruction manual for the body. Certain genes code for certain proteins, which in turn make up all the different parts of the organism. Any trait that you can think of is coded by DNA. Now, once you know what codes for what, you can use genetic engineering to essentially edit the parts you want, to some extent.
Genes are passed on to an organism's offspring so that its traits are inherited. Most biological traits are under the influence of many genes as well as the environment. Some genetic traits are instantly visible, such as eye color or number of limbs, and some are not, such as blood type.
Hey there! You may be wondering what you're doing at the front of a bioengineering lab and why a total stranger is talking to you right now. But I'm here to talk about some of the awesome experiments that Herbert Boyer and Stanley Cohen did in the 1970s. You might be thinking to yourself, the 70s? Isn't that like when the dinosaurs lived? Boring. Well, have you ever gotten a vaccine or eaten groceries from a grocery store? That's all thanks to some of the experiments that these two lab pals did in the 70s, where they took enzymes that cut DNA and were able to insert these pieces into new bacterial DNA. They pioneered the field of genetic engineering, and thanks to all the work that they did, we have medications for people with diabetes and heart attack victims. You know what, why don't you guys just follow me inside? So the first thing that Cohen and Boyer did was they took DNA from a frog and cut it up into pieces. I should probably explain to you guys what's going on in this tube. Here's some DNA from the frog. It encodes certain traits that affect how the frog develops, how it survives, how it looks. Now there's a piece of DNA in this genome whose job is to encode for certain proteins that the frog uses to survive. We're going to cut this piece out using a restriction enzyme. This is the thing that Boyer discovered. It's kind of like a pair of scissors. By cutting the DNA at specific points, we can get different pieces. Now, there are tons of different kinds of restriction enzymes, and they all cut in different places in DNA. Our enzyme that we're using today is called EcoR1. Now, this piece of DNA has more than one spot that EcoR1 can cut, as shown by the dotted lines. So we're going to end up with other pieces of DNA in addition to the one we actually want. Now, that's actually okay, because we're going to deal with that problem later. What's going on in the tube is we're literally just cutting up our piece of DNA, just like this. Now, here's a tube full of plasmid. It's basically a circular ring of DNA that can exist in bacteria, and it basically encodes for certain traits that affect how bacteria look or act or survive. Now, to add our DNA from the frog DNA to our bacterial DNA, we need to cut the plasmid open too. So we've got to add our EcoR1 scissors, and they're going to cut at this site. You let this reaction proceed, letting all the ingredients, the plasmids, the restriction enzyme, and some water and buffers to hang out together until all the plasmids are cut like this. Now we need to add the DNA pieces from the frog and allow the pieces to join our cut plasmids. Here's what's going on in the tube now. I've got pieces of frog DNA, and I've got cut plasmids like this. I need to add this guy into this plasmid. And the way I'm going to do that is by adding an enzyme called DNA ligase. This enzyme kind of acts like tape, and it fixes the DNA insert into the plasmid.
Applications in medicine. Genes are responsible to a large extent of who we are and how we behave. Behavioral genetics studies heritability of behavioral traits, and it is an overlap of genetics, psychology, and ethology. Developmental genetics, similarly, is a study of how genes influence behaviors. One kind of biotechnology is gene technology, sometimes called genetic engineering or genetic modification, where the genetic material of living things is deliberately altered to enhance or remove a particular trait and allow the organism to perform new functions. Genes within a species can be modified, or genes can be moved from one species to another.
Can we imagine a tomorrow when we'll truly be stronger than the diseases that separate us today? When we'll overcome the ravages of fatal threats with simpler procedures that cure the body and enhance the spirit? This day may arrive sooner than we think. When it does, a greater understanding of our genetics will be at hand. Every day, we see life moving faster than ever. We remember simpler times, but must embrace the possibilities of a bold future. Your lives, and those of all children, will change. A revolution in genetic engineering promises a new day for all of us.
We have the capacity and facility to change the very nature of humankind.
And there's a lot of debate about what you should allow and what you shouldn't allow. There's going to be a whole spectrum of things that are possible. A lot of it will be very uncomfortable with.
Pros. Using human genetic engineering, it could be possible to change the way genomes are constructed to end some diseases.
Vsauce, I'm Jake, and how would you like to be better? We don't get a say in what our genetics say in those 6 billion letters of DNA that make us, us. But what if we did? Now, we might not be able to adjust our DNA like you can in the video game series BioShock, where plasmids, gene tonics, and vigors allow you to enhance your genetics to such a degree that you're shooting out bees or electricity. But what about increasing your strength or your speed? Or your mind? Can you genetically enhance yourself? First, we should start by treating or preventing whatever illness we might already have, using gene therapy. Let's draw this one out with ASAP science.
Whether you have an IQ of 50 or 150, we all like to feel smart. After all, the minds of intelligent people like Einstein, Tesla, or Stephen Hawking have no doubt helped shape the world we live in. So, is it feasible that we could genetically improve the intelligence of our entire species? DNA is the basic code of life found in every living organism, and this DNA is made up of genes, which in turn are made up of 4 smaller parts called nucleobases, represented by the letters A, T, G, and C. In humans, these bases are arranged as 3 billion pairs, which essentially act as a blueprint for everything the cells in your body need to do. Literally all that you are, from the way you look and act, is defined by the organization of these 4 biological compounds.
And scientists have been able to find connections between the way genes are organized and certain traits you exhibit, from your sex to your eye color and even diseases you may develop.
It was actually the Human Genome Project which first began to analyze and map all the genes in the human body. The initial project took over a decade and $3 billion to complete, with teams across the entire globe working together. Today, with a much smaller team, the same could be accomplished in a few days for only $4,000.
And the more sequenced genomes we accumulate, whether it be from plants or microbes or humans, the more we have to compare and contrast the traits we see on the outside with the gene combinations we find on the inside. And that
Without certain diseases to increase death rates and decrease lifespan, it would be possible for more individuals to live longer and healthier lives. Having a genetic screening with a fetus can allow for treatment of the unborn. Over time, this can impact the growth and spread of diseases in future generations. Cons. Ethical issues. The belief that God should have ultimate power and we should not be altering nature is what many think should halt the progression of human genetic engineering. Safety issues. Getting genes into the human body is a complex process that could go bad very easily. The extent to the consequences if it goes bad are not fully known and could be quite devastating. The success rate is also something that brings up concern. Conclusion. The study of genes can bring great benefits. Because of ethic and safety issues, people are against it.